All right, guys, so this is Unit 2, and Unit 2 covers cell structure and function. For this unit, there are 11 topics in this unit, so this is a pretty long unit. For this lecture presentation, I'm going to be covering topics 2.1, which is cell structure and subcellular components, and then I'm going to go to 2.2, where we're going to be discussing the unique structures and functions of these cell components. And we're going to skip over a few of these topics, and then we're going to go to 2.10 and 2.11, where we're going to be discussing cell compartmentalization and the origin of cell compartmentalization. So we're going to be covering 4 topics for this lecture, and then we're going to go back and we're going to do the other topics. And trust me, it flows better this way. So here we go. All right, so let's start off with just a foundational definition of cells. So cells are the basic structural and functional units of all living things. And this is a part of the cell theory. And if you guys do recall, the cell theory is made up of 3 parts. And this is one of the parts of the cell theory. So the cell theory is pretty much, I believe, that all living things have cells come from pre-existing cells, and the third part of the cell theory is that cells are the basic structural and functional unit of all living things, which means that all the processes and all the characteristics of living things can be conducted by a single cell. So if you were to divide any living thing to its simplest form that still could continue life, that would be a cell. So let's talk about some of the parts or the functions that all cells possess, irrespective of how specialized those cells are, or what their specializations are, or what type of organisms these cells are found in. So all cells have a plasma membrane. Another name for plasma membrane, or another term for plasma membrane, is cell membrane. All cells have or is comprised of cystole. And cystole is the same thing as cytoplasm. All cells contain genetic information in the form of chromosomes. And we're going to talk a little bit about the structure of chromosomes in prokaryotic cells versus eukaryotic cells. And I believe that we did touch on this when we did our Unit 5, where we spoke about circular chromosomes as opposed to linear chromosomes. And the final and fourth component of all cells are ribosomes. So all cells do have ribosomes because all cells need to synthesize protein. Ribosomes differ in prokaryotic organisms versus eukaryotic organisms. However, their basic function is the same, and ribosomes are responsible for synthesizing proteins. So, what is a cell, and what are all the things that are common to all living cells? All right, so let's go to a quick comparison of prokaryotic cells and eukaryotic cells. So cells can be divided into one of 2 groups. A cell is either a prokaryote or eukaryote cells. So we call these cells prokaryotic when we're describing them, or eukaryotic cells. The organisms that possess the cells are called either prokaryotes or eukaryotes. So if an organism has a prokaryotic cell, then that organism is described as a prokaryote. All right, so let's quickly try to establish a comparison of the 2. Prokaryotes are bacteria and archaea. So these domains are comprised of prokaryotic cells. Prokaryotic cells do not have a true nucleus, but they do have a region where the genetic information is housed, and that region is called the nucleoid. So do not have true nuclei. So please remember that. Prokaryotes are generally smaller in size, so they're very simple cells, and this is also as a result of them just not having as many organelles, especially like cell membrane-bound organelles. All right, let's hop over to eukaryotes. Eukaryotes are pretty much all other living things that are not bacteria or archaea. So we have protists, fungi, animals, plants. So any other living thing that is not in the domain bacteria or archaea, those are eukaryotes. Eukaryotic organisms do have a true nucleus, so please remember this. So nucleus is found in eukaryotic organism, and the nucleus houses the genetic information in the form of linear chromosomes. So that means that the genetic information does have a beginning and it has an end. And eukaryotic organisms do contain cells that have several membrane-bound organelles, which means that these organelles or these subcellular components are surrounded by a membrane of some sort. All right, now we're going to start discussing some of these organelles. All right, key things when discussing organelles, we need to have like a unique description of the structure of that organelle, and also the function. So what does the organelle do? So what does it look like? What is it made up of? And what does it do? All right, so there are two categories for organelles. Um, Organelles are usually described as membrane-bound structures in eukaryotes, which means that these organelles are surrounded by a membrane. So they're pretty much enclosed. So they're pretty much cut off from the external environment. Even though materials can get into and out by various means of transport, but however, they're usually separated from the external environment that they're in, and that's as a result of the membranes that these organelles have. So what are the two classification of organelles? Organelles are described as endomembrane, which means that within the cell there is a membrane that surrounds these organelles. And here are some examples of these organelles. The nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, vesicles or vacuoles, and the plasma membrane itself. And then we also have another category of organelles. They do have membranes, so not to be confused, not saying that these organelles don't have membranes, but these organelles, they do serve a very specific purpose, and that is usually to like generate energy, or they're associated with energy. And the two organelles that are associated with energy is the mitochondria and the chloroplast. And these organelles we're going to be discussing in great detail when we get to Unit 3. All right, guys, so I'm going to highlight this region of my slide right here. And what this means is that I have now jumped to topic 2.10 because I want to just quickly explain to you guys what cell compartmentalization is before I start discussing the structures of the organelle. So cell compartmentalization is the ability of an organelle or organelles to allow for different metabolic reactions to occur in different locations of the said organelle. So in simple terms, if an organelle is conducting metabolic reactions, however, those metabolic reactions have specific places inside the organelles where those reactions will occur, then that means that the organelle is compartmentalized. And if you're confused about what a metabolic reaction is, a metabolic reaction is pretty much any chemical reaction that is occurring inside of a cell. Because cells have organelles that do carry out specific functions, there are organelles that are responsible for specific chemical reactions. So, for example, energy is generated in the mitochondria. How is energy generated? Through the process of cellular respiration. Cellular respiration occurs inside the mitochondria. However, there are different stages of cellular respiration, and each stage will take place in a specific region of the mitochondria. So the mitochondria is compartmentalized because there are different compartments inside of the mitochondria, which is an organelle, that allows for specific stages or specific types of metabolic reactions to occur. Now, why are these things important? Why is compartmentalization important? Or what is the purpose of compartmentalization? And here are two reasons why compartmentalization is important. What it does is it increases the surface area for these reactions to occur. And also, it prevents interfering or reactions from occurring in the same location. So if you're compartmentalizing different stages of the reaction, it means that these two reactions are not going to be interfering with each other. And those are the two main purposes or advantages of cells being compartmentalized. All right, so now we're going to discuss some components that are unique to plants and animal cells. This is a typical plant cell. This is a typical animal cell. So we use these diagrams for the sake of an example. These are not specialized cells. Plants and animal cells typically do not look like this because plants and animal cells would be specialized based on their functions and where in the organism they're found. All right, so let's start off with some components that are unique to plant cells and plant cells only. Plant cells have chloroplast, plant cells have central vacuole, plant cells have cell wall, and plant cells have pores like these ones that are called plasmodesmata, and this plays a role in cell-to-cell communication, and we're going to discuss that in Unit 4. Here are some units that are unique to animal cells. Animal cells has lysosomes. Animal cells have centrosomes. They have flagella, and those are the structures that are pretty much unique to them. They also have vacuoles. It's not listed here, but their vacuoles are smaller and they're larger in quantities. So they're usually very small, almost like dot-like structures within the animal cells, and they're described as being more plentiful, and also they're temporary vacuoles. So animal cells can lose their vacuoles. Plant cells have large central vacuoles, and their vacuoles are pretty much fixed. They never lose their vacuole. They can reduce in size depending on the amount of water that is in the vacuole, but it's usually just one central vacuole for a plant cell, and they don't usually lose their vacuole. However, animal cells have temporary small vacuoles. Let's go through the structure and function of some of our endomembrane organelles. So we discussed what these guys do or what they are in our previous slides. We didn't discuss the function, but here we go. We're going to start discussing their function. So we're going to start off with the nucleus, which is one of the most important organelles in the cell. Bear in mind that when we start discussing nucleus, if a cell has a nucleus, then it means that the cell is a eukaryotic cell, because those are the only cells that do have true nucleus. The nucleus is responsible for housing genetic information. So it contains chromosomes, which is genetic information. The nucleus is enclosed by what is called a nuclear envelope, and that's pretty much a nuclear membrane. And we did say that this is an endomembrane structure, so we're expecting it to have a double membrane. So here we go, double membrane. And these membranes, they contain pores. So on my diagram right here, I have these little holes or pores, and these are nuclear pores, and that is where the mRNA will exit after transcription, among other things. So also when the ribosomes are produced in the nucleolus, which is in the center of the nucleus, they also exit through those pores. So the pores can regulate the entry and exit of materials from the cell, and those are the pores. The nucleus contains another innermost structure that is called the nucleolus. It's a dense region of the nucleus where ribosomal RNA is synthesized. So ribosomes are made in the nucleolus, and the ribosomes make proteins. So our RNA is combined with proteins to form large and small subunits that are called ribosomes. And that's a key thing. So that's where we are expecting a ribosome or RNA to be made. Subunit exits. So these ribosomal subunits will leave the nucleus via the nuclear pores. And that's where this is pointing to the nucleolus, which is the center of the nucleus. What do ribosomes do? Ribosomes are responsible for translating messages coming from the messenger RNA, and they make the primary structure of proteins, which is pretty much a polypeptide chains. And we know all about this because we did this in Unit 6, and we did this also in our previous unit, which is... All right, now let's talk about these guys, ribosomes. So ribosomes are produced in the nucleolus of the cell, and the nucleolus is found in the nucleus, as we discussed in our previous slide. So ribosomes are made up of two parts. A ribosome is comprised of the rRNA and a protein, and ribosomes are usually called ribosomal subunits because there are two parts to those things. Ribosomes, the function of a ribosome is to synthesize proteins, so it produces proteins. Ribosomes can be found in two locations. Either they're going to be like free floating ribosomes, and those are usually going to be found in the cytosol, also called the cytoplasm. Ribosomes that are found inside the cytoplasm, they produce proteins that serve functions inside said cytoplasm or cytosol. And these proteins usually, not always, but usually they are enzymes. And remember, enzymes are a type of proteins. So if the ribosomes are not attached to any other organelle and they're free floating ribosomes inside the cytoplasm, also called the cytosol, then we're expecting that these proteins will serve functions within that cytoplasm. So they do things within the cytoplasm, or they carry out metabolic reactions. And these proteins are usually enzymes, and they speed up chemical reactions. Now, another place where you can expect ribosomes to be found is bound to the endoplasmic reticulum. And you should know that if ribosomes are found on the endoplasmic reticulum, it makes the surface of the endoplasmic reticulum bumpy, so it's not smooth, and therefore it is called the rough ER. So rough ER is as a result of ribosomes bounded to the surface. Now, if the ribosomes that are on the ribosomes that are on the ER, if they produce proteins, or when they do produce proteins, these proteins are usually secreted from the cell. So it means that these proteins go somewhere else, pretty much. So secretion means to leave. So it means that the cell is going to send these proteins somewhere else. So maybe they're messenger proteins, or maybe they're made to be sent to other parts of the cell, or other cells, or other parts of the organism. All right, so those are the key things about ribosomes. Oh, and also they leave by vesicles, and vesicles are pretty much just any membrane-bound structure that is used to transport substances. All right, so we mentioned the endoplasmic reticulum, so let's talk about that organelle. So the endoplasmic reticulum is a network of membranous sacs and tubes. So remember this description as for the structure. So it's membranous, and it is sac-like, and it is also tubular, so it's made up of sacs and tubes. The function of the endoplasmic reticulum. Before I go to function, I want to point something out, because oftentimes students do confuse the endoplasmic reticulum with the Golgi apparatus, not with the function, but with the structure. The endoplasmic reticulum is usually going to be found attached to the nucleus. So in a cell that contains the endoplasmic reticulum, it's usually going to be like extensions of the nucleus. And it's actually like extensions of the membranes of the nucleus. So this is our endoplasmic reticulum. So this is our endoplasmic reticulum. So if you're given a diagram in which you're asked to identify this thing and it's not labeled, look for nucleus, and wherever nucleus is where the endoplasmic reticulum is. It's going to be attached to the nucleus. It's almost like they're folds or extensions of the nuclear membrane, and that's why they're called membranous sacs. And we're going to discuss Golgi apparatus, but the Golgi is always going to be just by itself. It also has several folds or lobes. However, it is not attached to the nucleus. The endoplasmic reticulum is always going to be attached to that nucleus. So it's almost like an extension of the nucleus, like the nucleus membrane is folding out. All right, so that's the structure. Now let's talk about the function of the endoplasmic reticulum. It is responsible for, it synthesizes membranes for the nucleus, or it also synthesizes membranes for other organelles. It helps to keep the cell as a unit compartmentalized. So it helps to produce these. So remember, to synthesize means to produce. So it helps to produce these membranes so that organelles can have membranes, and by doing so, it keeps the cell compartmentalized. So it separates the organelles from pretty much the internal part of the cell, or it keeps the organelles separated from the cell. So organelles can be enclosed, or organelles can be compartmentalized themselves. There are two types of endoplasmic reticulum. So we have, and a short for endoplasmic reticulum is ER. So we have rough and we have smooth endoplasmic reticulum. The rough endoplasmic reticulum will be the one that contains the ribosomes, that synthesizes proteins that will be secreted by the cell. And the smooth endoplasmic reticulum is responsible for, is more involved with the lipid synthesis. All right, so endoplasmic reticulum. So let's look at the structure. So this one right here, we can easily identify this guy as being the rough one. And this is the smooth one. And remember what makes the endoplasmic rough and what makes the endoplasmic reticulum smooth. So the rough one contains ribosomes that are bounded to the membrane. So these ribosomes are attached. So once they're produced by the nucleolus inside the nucleus, they're going to leave through the nuclear pores and they're going to attach themselves to the endoplasmic reticulum. If the endoplasmic reticulum is smooth, it means that it does not have ribosomes attached. The smooth ones contain no ribosomes. What do the smooth ones do? They synthesize lipids. So please remember that. They synthesize lipids. They also metabolize, and remember, metabolism is any chemical reaction that occurs in the cells. So they're involved in chemical reactions that incorporate carbohydrates, and they're also responsible for detoxifying. So detoxifying means to get rid of toxins from the cell. So I've highlighted some things that you need to know about the smooth ER. In most cases, students can easily tell the function of the rough ER, but the smooth ER is sometimes where students kind of struggle a little bit. So smooth ER, these are the key functions of the smooth ER. And remember the structure, it is smooth because it does not contain ribosomes, and also it is membranous, it is an extension of the nuclear membrane. All right, so let's move on to the Golgi complex. So Golgi complex, also known as Golgi bodies or Golgi apparatus, so it's the same things. It's just three different terms for the same structure. I'm not sure why it has three different terms, but they all mean the same thing. All right, so let's start off with a description of the structure. The Golgi apparatus or the Golgi complex is made up of membranous sacs, very similar to the endoplasmic reticulum, and that's why oftentimes they're both confused, their structures are confused. However, these membranous sacs are flattened, so they're flat like sacs. And also remember what I said earlier, the Golgi complex is not attached to the nucleus, while the endoplasmic reticulum, they are attached to the nucleus and they're extensions of the nuclear membrane. So here's also another key thing to note with the structure. These flattened membranous sacs are called cisternae, so that's their name. That's the name that is given to these sacs. And each individual sac is separate, unlike the endoplasmic reticulum. Um, so in between, so even though they're very close together, there is cytoplasm or a cystole between them. So they're very tightly packed, however, they're not attached. Each cistern is not connected. So that's what I just said. And then also, there is some directionality to the Golgi apparatus. The Golgi apparatus, its function, and I'm going to get into its function in a lot more detail, but it is regarded as the UPS of the cell. So it is responsible for packaging and processing protein. I should have said this earlier, guys. You guys might know several analogies for cells. Please do not use analogies as definitions. Do not use analogies as examples. I would advise that you don't use analogies at all ever on your AP exam, because analogies are not accurate functions or descriptions of the structures of these membranes. So we just use them for you guys to remember or to liken them to something that you already know, but these do not suffice as definitions for any of these organelles. All right, so the Golgi apparatus is responsible for pretty much for processing and distributing proteins and modifying proteins. However, there are different regions that do different things. The Golgi complex will be receiving and also distributing. So think of it like a distribution center. There is a cis phase, and the cis phase is responsible for receiving vesicles from the ER. So anything that's coming from the ER will come to the cis phase. And then there is a trans phase that is responsible for pretty much sending things out, or yeah, pretty much sending things away from the Golgi complex. And I'm gonna use a different color, and I'm gonna show you where each things are. This is, and I'm gonna try to just draw a line here. This region right here in the front, where the smallest lobe or the smallest sac is, that is our cis phase. Our transphase is going to be behind where the longest flattened membranous sac is. That is our transphase. So again, the cisphase is responsible for receiving vesicles coming towards the Golgi complex from the ER, and the transphase is responsible for sending vesicles into the cytoplasm or the cystole to be distributed to other areas or to be secreted out of the cell. That was mostly a description of the structure. Now we're going to talk about the function. So what is the Golgi complex responsible for doing? It's responsible for receiving vesicles with materials that are coming from the ER, and by ER we mean endoplasmic reticulum, whether the rough or the smooth one. And it pretty much modifies these materials. Now, what materials would you expect to be coming from the ER? If it's a rough ER, you're expecting proteins to be coming in, and if it's a smooth ER, you're expecting lipids to be coming in. Now, it is responsible for modifying these materials. Specifically, one of the major roles of the Golgi apparatus is to modify proteins. So it changes the proteins. And also it is responsible, it's not on my slide here, but it's responsible for ensuring that the proteins are properly folded. So these proteins coming from the ribosomes. Remember that when proteins come from the ribosomes, guys, the ribosome is pretty much assembling the polypeptide chain. It has not folded into its secondary or its tertiary structure, or if it's going to be folded into quaternary structures. So that folding or that modification of the protein will occur inside the Golgi apparatus. So a long chain of polypeptides leave ribosomes. Ribosomes are found on the ER, and they come to the Golgi apparatus for modification. What are these modifications? Folding of the proteins, and sometimes adding other molecules to the protein. So for example, a protein can be modified into a glycoprotein by adding carbohydrates to that protein. So, and it also sorts other materials, not just proteins, and it adds tags to these molecules. So some of these cell tags that are responsible for cell communication are actually tagged in these Golgi apparatus. So they're like the packaging or the distribution center of the cell. And then also, once they're done, they will distribute these packages through their distribution regions. Remember, all right, so let's talk a little bit about lysosome. Lysosomes are very basic in terms of their structures. So key thing we need to know about the structure is that these are membranous sacs. So they're sac-like structures, very similar to vesicles. And inside these sacs, we're expecting to find hydrolytic enzymes. So they're sacs that are membranous, so they're bounded by cell membranes. Specifically, they're double membrane structures, so there are two membranes on the structure, and these organelles are responsible for speeding up reactions. How do we know they speed up reactions? Because they contain enzymes. What type of enzymes? Hydrolytic enzymes. When you think of hydrolytic enzymes, these enzymes, they speed up hydrolysis. And if you recall from Unit 1, hydrolysis is a condensation reaction in which macromolecules are broken down by adding water. All right, so now let's go into the functions. So they hydrolyze macromolecules in animal cells. And remember, these guys are found only in animal cells. So plant cells do not have lysosomes. They can also carry out a process that is called autophagy. And autophagy is pretty much when they break down themselves in order to recycle. So lysosomes can recycle their own cells, organic materials. And that helps the cells to reuse materials and also to renew itself. There's another organelle that is rarely mentioned, but it functions similar to ribosomes, and these are perioxysomes. And I remember teaching pre-AP Bio, and we did a cell campaign, and most of the students, that's the first time they're even hearing about this organelle. But this is not a very well-known organelle, but it is also membranous sac, and it specifically metabolizes a specific reaction. So let's talk about the function of perioxysomes. So they're similar to lysosomes in their structure. They have membranes, so they're membrane-bound organelles, and they carry out a specific metabolic reaction. they catalyze, and the word catalyze means to speed up. So they speed up the reaction, or a reaction that produces H2O2. And I want you to pause and think about what is that. And if you don't know, H2O2 is hydrogen peroxide. So peroxisome, hydrogen peroxide. And what they do is enzymes in these peroxisomes, and remember, they're very similar to lysosomes, so they do contain enzymes. They break down hydrogen peroxide to form water. And that's pretty much their sole function or purpose, is is pretty much to catalyze that specific reaction. All right, so let's talk about vacuoles. So vacuoles are pretty much vesicles, very large vesicles in some cells that are responsible for transport. The picture that you're seeing on your screen is depicting a vacuole that is found in a plant cell. And we know that it's a plant cell because the vacuoles in plant cells, they get very large. They are the largest organelles in plant cells, and they are described as central vacuoles. If you're ever given a picture of a plant cell and an animal cell, well, you should know what the difference is. But if it's not labeled and you're trying to identify which is a plant cell, which is an animal cell, is a key thing is to look for the vacuole and compare that to the size of the nucleus. If the vacuole is larger than the nucleus, then that is a plant cell. So vacuoles, they stem from the ER and Golgi apparatus. They're not attached to the ER or the Golgi apparatus, but they do stem from those. And they are pretty much, they play a role in transport, and they also play a role in storage. So what are the types of vacuoles? There are three types of vacuoles. We have food vacuoles, and these guys are responsible for storing food, which are useful substances for the cells. However, they're formed by a type of active transport that is called phagocytosis, and this is also referred to as cell eating, in which a vesicle takes in pretty much solid substances, and these solid substances can further be broken down by lysosomes. And remember, lysosomes are enzymes, they're organelles that contains enzymes. Here's a second type of vacuole, not found in all living things, specifically found in paramecium, which is a type of protist, and protists are single-cell organisms. So parameciums, they have these type of vacuoles that are called contractile vacuoles. And they're called contractile vacuole because they can contract and relax to pretty much squeeze water out of the cell if needed. And as a result of doing this, they maintain the water level of the cell. So they play a role in osmoregulation of the cell. And then we have central vacuole. Central vacuole is found in all plant cells, and these are the big organelle that is found inside the plant cells. And what do central vacuoles contain? A lot of water and also inorganic ions, and they play a role in turgor pressure of the cell. So they pretty much play a role in keeping the cell osmoregulated as well. And that's a picture of the central vacuole. All right, so we're almost there, guys. We're almost done discussing organelles and their functions and structures. All right, so there are two more organelles that we're going to be discussing, and these two organelles, they're classified as energy organelles. Before I start discussing these two organelles, I want us to the endosymbiont theory is what we're about to discuss. All right, so I want to highlight the bottom of my screen right here. So this is for you to know that, hey, I've jumped to 2.11, which is the last topic in Unit 2. However, I think this is a good time to introduce that topic. So in that topic, we're going to be discussing the endosymbiont theory. And I'm going to go ahead and I'm going to break this word into two. Endo means in, symbiont means theory. And this is a theory that seeks to explain the similarities between the mitochondria and the chloroplast to prokaryotes. So in simple terms, this is a theory that explains why is it that mitochondria as an organelle and chloroplast as an organelle is so similar to prokaryotes as living things. And here's what the theory states. The theory states that an early eukaryotic cell So pre-existing many, many millions of years ago, eukaryotic cell or a eukaryotic cell engulfed or took in a prokaryotic cell. So the prokaryotic cell became an endosymbiont of this eukaryotic cell, which means that the prokaryotic cell now lives inside the eukaryotic cell. And they became one functional organism, right? So that's pretty much the endosymbiont theory in just in a nutshell. All right, what are the lines of evidence for this? Now, bear in mind that the endosymbiont theory is seeking to explain why is it that the mitochondria as an organelle and the chloroplast as an organelle is pretty much similar to prokaryotes as organisms. So there are a few things that we need to know about the mitochondria and the chloroplast, and we're going to be digging a little bit deeper into those because we're going to be discussing those as organelles. Um, Mitochondria and the chloroplast are double membrane structures. So is a prokaryote. So prokaryotic cells or prokaryotes as an organism is also double membrane. Prokaryotes have ribosomes that produce or synthesize proteins. A mitochondria and a chloroplast also has ribosomes that synthesize proteins. So in addition to the ribosomes that are found in the cells in general, the mitochondria and the chloroplast as an organelle has its own ribosomes inside that synthesizes its own proteins. So the mitochondria and chloroplast do not rely on the ribosomes inside the cells to produce proteins for them. Also, if you didn't know, the mitochondria contains genetic information, and also the chloroplast contains genetic information, and the genetic information is in the form of circular DNA. You know what also has circular DNA? Prokaryotes. And the mitochondria and the chloroplasts are capable of pretty much living on their own. So they're capable of functioning on their own without the cell. So even though these are organelles within the cell, they're independent organelles within cells that can pretty much carry out most of the functions that they need for their own survival without depending on the cell. And that pretty much, these are the lines of evidence that, hey, maybe the mitochondria was a prokaryotic cell. Maybe chloroplasts were prokaryotic cells that were engulfed by a eukaryotic cell. However, they have maintained their true characteristics and they have maintained their independence pretty much even though they are functioning as one. So they have aided eukaryotic cells to evolve because the mitochondria is producing energy and the chloroplast is able to photosynthesize, so it captures sun energy, produces its own food. However, these are independent organelles of the cells that they're living in. So they're endosymbionts of these cells. However, they're still independent organisms of the cells. All right, so now we're discussing the mitochondria. And the mitochondria is an energy organelle. And I want to pause here a little bit, guys, and I'm trying not to make these videos too long. I already failed, but I'm going to pause right here. And I just want to say, when you guys are discussing the structures of these organelles, try to recall or always try to use structural descriptions that are unique to the organelle that you're trying to describe. So, for example, if you were to say that the mitochondria is a double membrane structure, that is correct. However, that is not unique to the mitochondria. So, if you're ever asked a question in which you're to describe the structure of the mitochondria, put descriptions that are unique to the mitochondria that only the mitochondria has, and do that for any organelle. So always try to find structures that are unique to that organelle when you're doing structural descriptions. All right, so let's talk about the mitochondria. The mitochondria is where cellular respiration occurs. So that is where energy is generated for the cell. And we're gonna get back to the function in a little bit, but let's go to this description. The mitochondria is a double membrane organelle. Remember that it is, that's true, but however, that's not unique to the mitochondria. Here are some structural uniqueness that the mitochondria has. The mitochondria has an outer membrane that is smooth. Here is the outer membrane, smooth outer membrane. It also has an inner membrane that has folds. Here is the inner membrane. The inner membrane has folds, very unique to the mitochondria. The folding of the inner membrane is called the cristae. So that inner membrane, as a result of those foldings, they're called cristae. This divides the mitochondria into two internal compartments. The mitochondria is compartmentalized. The compartmentalization of the mitochondria increases the surface area for metabolic reactions to occur. Now, as a result of having an inner and an outer membrane, so smooth outer membrane and folded inner membrane, it creates a region that is called the intermembrane. So the intermembrane is that region or that space in between the inner and the outer membrane. Okay, so still on the mitochondria, describing unique functions. The mitochondria has a region that is called and this is enclosed by the inner membrane. So again, remember the inner membrane is the one that is folded. The inner membrane goes all the way around, and inside that folded inner membrane, this inner region right here is called the mitochondrial matrix. And that is where the Krebs cycle occurs. And the inner, sorry, or the mitochondrial matrix, this region right here, it contains enzymes that catalyze cellular respiration and also produces ATP. Very important things. I'm about to state two very important things. In this mitochondrial matrix is where the mitochondrial DNA is housed, and also that is where the mitochondrial ribosomes are housed. And remember, we discussed the endosymbiont theory. Mitochondria has its own DNA, similar to prokaryotes, and the DNA is not in the form of linear chromosomes, but instead it's circular chromosomes. Mitochondria has its own ribosome, which means that it synthesizes its own protein. It does not depend on the cell's ribosomes to synthesize proteins for it. And those two things, those two additional organelles are found inside the mitochondrial matrix. All right, so as I mentioned earlier, the mitochondria is the site for cellular respiration. So this is where energy will be synthesized. There is a correlation between the metabolic activity of a cell and the number of mitochondria that the cell has. If a cell has a high number of mitochondria, then we're expecting that cell to have a high metabolic activity. So the cell is going to be doing a lot of work if it has a lot of mitochondria because it requires a lot of energy. So cells with high metabolic activities have more mitochondria. Therefore, cells that have to move or contract will have a lot more mitochondria than cells that pretty much don't move, they don't contract, and they don't require a lot of energy. All right, we're almost done, guys. So we're now on the chloroplast. So the chloroplast is another energy organelle, and chloroplast is found only in plant cells and some bacteria. So, like for example, they're found in cyanobacteria, and those are bacteria that can photosynthesize. Chloroplast is associated with photosynthesis or photosynthetic activities, and this is what gives any organelle that possesses a chloroplast the ability to pretty much make its own food, and that organelle is classified as a photoautotroph. All right, so chloroplast, which is an energy organelle, is specialized organelle in photosynthetic organism, and this is a site of photosynthesis. The chloroplast contains green pigments that are called chlorophyll, and there are different chlorophyll pigments, and we're going to get into that when we do photosynthesis. It is also a double membrane. So we're now describing the structure, and as I said in my previous slide, yes, it is a double membrane, but this is not what makes the chloroplast unique. It is a valid description, is a correct description, but if you were to give a description of the chloroplast, if you were to ask to give a description of the chloroplast structural description, avoid using this and use descriptions that are unique to the chloroplast. And if you do use double membrane, accompany that with some unique descriptions of the structure of the chloroplast. So here are some. The chloroplast contains thylakoids. What are thylakoids? These are membranous sacs that are organized into stacks called grana. So they're stacked on top of each other like a pancake. So that's what the thylakoid looks like. And what occurs in the thylakoid? The light-dependent reaction. So this is the part of photosynthesis that actually relies on sunlight. And here they are. That's what it looks like inside the chloroplast. And that's what it looks like stacked together to form the grana. Also, other regions, structural regions of the chloroplast is the stroma. And the stroma is not a physical structure, it is more like a space that is filled with fluid. So it's a fluid-filled space um, that's pretty much surrounds the thylakoids. And this is the location for the Calvin cycle, also called the light independent reaction. Um, this stroma also contains chloroplast DNA. And remember, we said from the endosymbiont theory, the chloroplast has its own genetic information. Also, this is where we're expecting ribosomes to be found. And again, ribosomes are found in chloroplasts, which means that they synthesize their own proteins. There are various enzymes that do play a role in the Calvin cycle or just photosynthesis in general, and those enzymes are going to be found in the stroma as well. All right, so here are some other regions that are more structural regions for structural support. These are not energy organelles, so we're going into another category. So these organelles, they do play a role in structural support. So we have the cytoskeleton. This is a network of fibers throughout the cytoplasm, and it gives structure and support to the cell, especially for animal cells, because remember, animal cells do not have a cell wall. So structural support is particularly needed for animal cells. It also provides some mechanical support, and in mechanical support, some physical support that assists in moving organelles around and also anchoring those organelles inside of the cell. So as I said, it allows for movement of vesicles and organelles as a whole. Movement occurs when the cytoskeleton interacts with other proteins called motor proteins. And when you think motor, you should think movement. So those proteins actually assist with movement. These are fibrous structures. The fibers in the cytoskeleton are made up of microtubules or microfilaments or intermediate filaments. So these are the different types of fibers, and we're going to get into all of these. So starting with the microtubules. Microtubules are like rod-like structures, so they look like little rods or sticks or like pasta before they cook, like little noodles. So these are hollow rod-like structures, and they're made up of a type of protein that is called a tubulin proteins. They grow to form what are called centrosomes. And centrosomes are very important structures that do assist with, especially in the process of mitosis and meiosis. They assist in the microtubules in assembling. Their function, they serve as structural support, and they pretty much help to move organelles along. And as I said earlier, they do function in mitosis and meiosis when chromosomes need to be separated or divided during the various PMAT phases. So prophase, metaphase, anaphase, telophase. And they also help to move cells as a whole. So they give cell motility by moving on the inside, pushing against the cytoplasm and just propelling the cells forward. They also form cilia and flagella, and these cilia, one cell will contain several cilia that help to move, or it moves in beat-like patterns to pretty much get rid of dust particles. So like for example, the cells that are found in the lungs or lining the respiratory system, those are made up of cilia, and flagella are found in like parameciums, or some amoeba have flagella, and also you have sperm cells that also have flagella. All right, now another cytoskeleton is microfilaments. These are thinner rod-like structures, and these are made up of protein, so they're not hollow. And the protein that they're made up of are called actin. Their main function is to maintain the shape of the cell, and they also give some tension to the cell. And in muscles, they will assist in muscle contraction and the movement of muscle cells. And if you guys do anatomy and physiology next year, or whatever year you guys do anatomy and physiology, if you choose to do this, we're going to learn a lot about the structure, the anatomy, and the physiology of muscle cells. Actin works together with another protein that is called myosin. And whenever you see myosins, then you should always think muscles. And they do help the muscles to contract and relax in order to move those muscle cells. Microfilaments can also play a role in the division of animal cells. So during the mitosis and meiosis phase, they are responsible for forming that cleavage furrow. So specifically during telophase, when the cell starts to separate. Intermediate filaments is the last one we're going to be discussing. These are fibrous proteins, and they're made up of varying subunits. So there are various components to this. These are permanent structural elements of the cells. So they die with the cells. They're not assembled or broken down at the rate at which the microtubules and the microfilaments are broken down. So thus why they're considered to be permanent. Their function is pretty much to maintain the shape of the cell. They also anchor the nucleus and the organelles, so they keep those organelles in place, and they form the nuclear lamina. So that is like the central region or the holes within that nucleus. So it lines the nuclear envelope, and they pretty much give structural support. Here are some questions, and I don't want to make this video too long, but the questions are here. We're going to be practicing several questions in class, but here are some questions that you guys, the answers are already exposed to you guys. So this is just like practice. And this is all she wrote for unit 2.1, point 2, point 10, and point 11.